So this brings us to a comparison between aerobic with anaerobic respiration. Remember comparison, we look at the similarities and the differences. So the similarities between aerobic and anaerobic both lead to release of energy, occur in living cells, release carbon dioxide. In both ATP is used. Remember, ATP is used to form ATP. Therefore, in both there is release of ATP, but in both there is even the use of ATP. So those are the similarities, just like we have earlier noted. The differences are also very much clear, whereby one occurs in presence of oxygen, anaerobic in absence of oxygen. Which one yields more energy? Aerobic. Anaerobic yields little energy. In aerobic, we have the glycolysis, Krebs, and electron transport. Anaerobic, we only have glycolysis. Where do we have complete breakdown of glucose molecule? Aerobic. But in anaerobic, it is partial breakdown. Aerobic respiration, where does it occur? Involves mainly the cytoplasm and the mitochondria. While anaerobic, because there is no Krebs cycle, there is no ETS that occur in the mitochondria. That means anaerobic occurs only in the cytoplasm. Aerobic is longer, anaerobic is shorter. Aerobic occurs throughout the life of the organism, anaerobic only temporarily, and other differences, just like we are noting there. We now move on and see alternative respiratory substrates. What do we mean? We noted that the main supply of energy in the body is glucose. Even in glycolysis, we saw that starch and glycogen get converted to glucose. Therefore, carbohydrates generally are the main source of energy. Carbohydrates stored as starch. When you look at this flowchart, look at that part where we have carbohydrates. Carbohydrates, mainly starch in plants, glycogen in animals, get converted to glucose, which glucose undergoes those changes, remember them in glycolysis, leading formation of pyruvic acid, giving us acetyl-CoA that is fed into the Krebs cycle. But when we talk of alternative respiratory, we mean if carbohydrates are not broken down, then what else can be broken down to release energy? We talked of fats or lipids and proteins. We begin with fats. Fats, we look at our cycle where we are having fats. Fats are broken down in presence of an enzyme known as lipase enzyme. Lipase catalyzes the breakdown of fats into fatty acids and glycerol. Into fatty acids and glycerol. Now what happens to glycerol? Glycerol is converted to glyceraldehyde 3 phosphate, also known as the triose phosphate. Remember, glyceraldehyde 3 phosphate or triose phosphate is an intermediate within glycolysis. When fructose 1,6-bisphosphate, a 6-carbon compound, is broken down into 2 3-carbon compounds, each called glyceraldehyde 3 phosphate, and then glycolysis proceeds. Meaning glycerol is converted into glyceraldehyde 3 phosphate or triose phosphate, and then fed into the glycolytic pathway. That is why when we look at this one here, look at where we have fat. We see that fats are broken down into fatty acids and glycerol. But glycerol is converted into phosphoglyceraldehyde, PGAL. PGAL, also called glyceraldehyde 3 phosphate, also called triose phosphate. and then it enters the glycolytic pathway. It is fed into glycolysis. But we also have fatty acids. Fatty acids, for them, they do not enter the glycolytic pathway, but they undergo what we call beta oxidation. Beta oxidation, B oxidation, means, remember, fatty acids have long hydrocarbon chains. Now, the long hydrocarbon chains of fatty acids are broken down into shorter hydrocarbon chains, usually the ones that contain 2 carbon atoms. The long fatty acids, those hydrocarbon chains, each chain is broken down into shorter chains, each chain of 2 carbon atoms, a process we are calling the B or the beta oxidation, leading to formation of acetyl. Remember, acetyl is a 2-carbon compound. So when those long hydrocarbon chains are broken down into shorter chains, each of 2 carbon, we form acetyl that combines with coenzyme A to form acetyl-CoA. Acetyl-CoA. That is why here we see that fats broken down into fatty acids and glycerol. Glycerol enters the glycolytic pathway as PGAL, or as TP, or as GALP, while the fatty acids, when they undergo beta oxidation, I've explained what it is. They form acetyl that combines with coenzyme A, thereby forming acetyl-CoA. Remember, acetyl-CoA is within the mitochondria. But glycolysis occurs within the cytoplasm. So that is for the fats. How about the proteins? Now, proteins, like we know, that their building blocks are the amino acids. Equally, proteases or protease enzymes catalyze, speed up the breakdown of proteins into amino acids. Remember, for fats, we talked of lipases. Proteins, we are talking about proteases. They catalyze, they speed up the breakdown of proteins into amino acids. Amino acids then undergo what we call deamination. Deamination, the amino group is removed. The amino group is removed. We can see the NH2 there. The amino group is removed from the amino acid, a process we are calling deamination. Now that amino group is then passed out of the body, either as urea, or as uric acid, or as ammonia, depending on the organism. Now what remains after removing that amino group is then converted into pyruvic acid. It can also be converted into acetyl-CoA. I repeat, proteins broken down by protease enzymes forming amino acids, which amino acids then undergo deamination. Deamination removing the amino group, which is lost either as urea, ammonia, ammonia, uric acid, depending on the organism. Then what remains is used to form pyruvic acid or to form acetyl-CoA. When it forms acetyl-CoA, it is then fed into the Krebs cycle, thereby generating energy. Therefore, when we talk of the alternative respiratory substrates, we are basically referring to the fats and proteins. The fats we've seen hydrolyzed into fatty acids and glycerol, and the details just like we have explained. The proteins we've also seen broken down by protease enzymes forming amino acids that undergo deamination, then the residual compound enters the pathway mainly as pyruvic acid or acetyl-CoA, or it may enter as oxaloacetate or alpha-ketoglutarate, We shall stop there for today. When we meet again next time, we shall see the last part of this topic called respiration, and that will be respiratory quotient, RQ. So that will be the last part of our topic, respiration. Until we meet again, God bless you all. Please keep safe. We need you alive. Remember, COVID is real.